All right, welcome everyone. Thank you all for coming today. My name is OccHealTSci. I'm a, what? Oh, well, okay. I'm an affiliate faculty member of the Institute, housed over at the School of Nursing, and it is my great honor, pleasure, everything to introduce Dr. Brooke Schaefer today to speak with us about a little bit about her data that she's worked on in our lab. She's been in my lab since 2023, joining us as a postdoctoral fellow, and she has been incredibly productive, and she is one of the best postdoctoral fellows I've ever had. She's also the only postdoctoral fellow I've ever had, but I think that ranks pretty high. She's been incredibly productive. She's written numerous manuscripts, received numerous grant awards. In fact, I believe she was telling me yesterday that when she joined the lab, I had her writing a grant within the first week of joining the lab, and she got that one. So she was on a T32 right away, and has also received the prestigious Sleep Research Society Foundation New Investigator Award last. So she'll speak to us about a little bit about those data and some of her past work and what she's looking forward to as she's just received the Birch K-12 Award. So very productive, and I can't wait to hear what she has to say. So help me welcome Dr. Brooke Schaefer.
Thank you. I'm kind of blushing, though. Such a nice introduction. So thank you, Dr. McHill, for the great introduction. Thank you for the seminar organizers for inviting me to talk today. I am very excited to share with you some of the stuff that I've been working on the last couple of years. But before I dive into that, I wanted to give a brief background just about myself, because I know many of you know me. Yes! 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 Amazing! Oh, this is going to be a great talk already. Yeah, double Duke here. So I did my undergraduate and my Master's of Science at James Madison University in Harrisonburg, Virginia. I focused my work in exercise physiology, particularly clinical exercise physiology, when I moved into my master's degree. My master's thesis data looked at patients in a cardiac rehabilitation program who had had previously undiagnosed obstructive sleep apnea. And then I took a chance and I moved to Canada for my doctoral training. I got my PhD at the University of British Columbia in Kelowna, BC. My degree was in kinesiology as well, but my research focused on hypertension development that was associated with obstructive sleep apnea. And so specifically how the hypoxia experienced during airway obstructions altered autonomic and vascular control, ultimately leading to hypertension. So as you can see, I was fortunate enough to pick two universities that had really bright and colorful cap and gowns. I was a little on the fence about the bonnet shown here from UBC, but I think I pulled it off okay. And then, as Andrew said, I joined the lab in the School of Nursing in 2023 and really embarked on a new adventure and into a whole new field of sleep and circadian research. And while my work up until this point had focused on obstructive sleep apnea, which is a sleep disorder, I wasn't necessarily looking at how that sleep disorder affected sleep. So I thought it might be kind of a fun visual representation. I took the words from my previous publications and threw them into a word cloud generator. And so we can see the big ones up until this point was sympathetic, hypoxia. A lot of my previous work had been in a male population. And you can see right here, sleep was a really tiny, tiny contributor. I think it showed up 3 times in the titles. So really can't stress enough how new I am to this field. And truthfully, I don't think I knew what a circadian rhythm was until a couple years ago. So I was really interested and really excited to find out that humans have these internal biological clocks that have evolved to anticipate fluctuations in light and temperature that occur as a result of Earth's 24-hour rotation. And so these endogenous mechanisms are called circadian rhythms, which are derived from the Latin circa, meaning approximate, and dien, meaning a day. Now, while these circadian clocks are anchored by genetics, daily circadian rhythms are synchronized or entrained to external time cues, with light being a particularly strong synchronizer to the 24-hour day. So light enters the retina and electrical signals get passed down to the suprachiasmatic nucleus in the hypothalamus, which is where our central clock is really located. And from there, the central clock aligns downstream clock-dependent gene expression and thus drives various physiological processes and behaviors. So some of the behaviors in humans include our sleep-wake cycle. So for us, that typically responds to us being awake and active and eating during the day, and asleep and resting and fasted at night. These behaviors are in part mediated by the hormone melatonin, which is involved in synchronizing these circadian rhythms and is the gold standard marker of circadian phase. And so as we can see here in this middle panel, circulating levels of melatonin are low during the day, begin to rise a couple hours before sleep, and then peak in the early part of the night. And we can actually measure this rise in melatonin onset in humans through a series of saliva samples. These samples are collected in a dimly lit room because light has a suppressing effect on melatonin production. And so this point at which melatonin really starts to take off is our dim light melatonin onset. And it's a nice way for us to assess how an individual is aligned to that 24-hour day. But with the advent of electrical lighting, we can now shift our school and our work activities into the biological night when our internal timing system is trying to promote sleep. So this causes a misalignment between our internal timing system and our behaviors, which can have adverse consequences to overall health. And so that's really what my work has been focused on since joining the lab a couple years ago, is trying to piece together how does this circadian misalignment affect health outcomes in various settings. And so one population that's particularly vulnerable to extreme circadian misalignments are individuals who are engaged in shift working occupations, where now they are awake and they're active and they're eating during the night and attempting to sleep during the day. And indeed, long-standing shift work is associated with up to a 40% increased risk of cardiovascular disease compared to individuals who work on a day-oriented schedule. And so one important indicator of future cardiovascular risk is the 24-hour blood pressure pattern. And so we see here that blood pressure follows a really nice circadian rhythm where blood pressures are elevated during the day and then they dip really nicely while we're sleeping. They dip below these daytime levels. And in individuals who don't have this substantial dip in blood pressure while they're sleeping are at an increased risk for future cardiovascular events and early mortality. So then the question becomes, what happens to this blood pressure dipping pattern in shift working occupations when now you have kind of this inversion in your sleep-wake cycle? So this was work done before I arrived in the lab by my mentor, Andrew Mackhill, looking at the acute effects of shift work on this blood pressure dipping response. And so in this study, they were able to look at baseline measures in newly hired bus operators during their day-oriented onboarding training. And then after their onboarding training, these individuals were then assigned their working schedule, with the majority of individuals being assigned a shift working schedule, while some of them still remained on days. And then these measures were repeated again at the 30-day mark, and then again at the 90-day mark. So really looking at those acute changes of what's occurring right when you're transitioning into a new job. So with this graph, we have the changes in blood pressure dipping. So that's demonstrated here on the y-axis, going from 0 to -20. This horizontal line would denote no change in dipping. So the definition of blood pressure dipping would be a greater than 10% reduction in blood pressure compared to daytime values. And so below that -10 would be a dipping profile, and above that -10 would be a non-dipping profile. And then on the x-axis, we have our time. So baseline, 30 days of shift work, 90 days of shift work. Individuals in the black would be the day-oriented schedule, and then the individuals in the red would be the night shift schedule. I realize I can't come too far away. So at baseline, there was no differences in blood pressure dipping between the two groups. We also see that there's no significant differences between these two groups at the 30-day mark. However, by 90 days after transitioning into a shift working occupation, we see that all the individuals that were assigned that night shift schedule now became non-dippers and had this non-dipping profile. And so there are rapid changes to blood pressure dipping that occur once starting this new shift working occupation. And so the next question would be, well, are there chronic effects? Are these adverse outcomes with shift work blunted or attenuated in any sort of way the longer that you are involved in the shift working job? And so this was the first project that I embarked on starting into the lab. And so we had 20 participants in this sample, 18 of which were female. These were all healthy full-time nurses, 10 of which were full-time day shift, 10 of which were full-time chronic night shift workers, with the average night shift duration being about 9 years. And then there was no self-reported history of hypertension, diabetes, sleep disorders, or pregnancy. Participants came into the lab and circadian phase was assessed by dim light melatonin onset. So briefly, participants spent the evening in a dimly lit room where saliva samples were taken every 30 minutes. And this allowed us to capture that point in time in which melatonin really started to rise before bed. Following the in-laboratory visit, we recorded 24 to 48 hours of ambulatory blood pressure readings. And then we assessed sleep onset by self-reported sleep diaries for a week after the in-lab visit. Our calculated measures included overnight blood pressure dipping, which is the ratio between sleeping and waking systolic blood pressure expressed as a percent. We also looked at the standard deviation of sleep onset timing. And so on a day-to-day basis, how variable, consistent are you going to bed between days? And this might give us an indicator of potential disruptions between our circadian system and our behaviors, right? If we're very variable, going to bed at different times throughout the week, that might indicate some circadian disruptions could be occurring. And then we calculated phase angle of entrainment, which is a way to assess the relationship between our internal biological clock, which in this case would be our DILMO, and an external time cue, which in this case would be our sleep onset. And so the time duration in hours between these two time points would be our phase angle, with a shorter time duration if these two time points were occurring closer together, typically being associated with more sleep and circadian disruptions. So if we look at our participant characteristics, we had no differences between the day shift and the chronic night shift workers in terms of age, BMI. There was no differences in waking, daytime blood pressure, either systolic or diastolic, and no differences in resting heart rate. But if we look at our sleep and circadian phase factors, we see that not only did the chronic night shift group have significantly worse overnight blood pressure dipping compared to the day shift, but they were also considered to be on average having a non-dipping blood pressure response. Furthermore, the chronic night shift workers had significantly later DOMOs, significantly higher variability in their sleep onset timing, and they had significantly shorter or narrower phase angles. And so here we are looking at the relationship between overnight blood pressure dipping and circadian alignment. We have our blood pressure dipping here on the y-axis and our phase angle of entrainment here on the x-axis. We may think that the shorter or narrower phase angles are associated with less circadian alignment, and the longer or wider phase angles would be associated with more circadian alignment.
Can I ask a technical question?
Sure.
So for the measuring the sleep and the melatonin for the night shift workers, when did you do that? I mean, if there wasn't a night shift, like when am I doing the night?
Yeah, that's a great question. So for everybody online, the question was when during their work schedule did we do the melatonin collection? It was during their off days. So I believe they had at least a couple days off in a row when we did those samples. So it wasn't like in the middle of their working schedule. And then same thing with the outpatient blood pressure. We did it during their off days. Great question. Thank you. So similarly to the graph before, the day shift is going to be in black and the night shift will be in red. And what we see is, regardless of day or night shift group, there was a significant inverse relationship between phase angle and our blood pressure dipping, such that a shorter or narrower phase angle was associated with poorer blood pressure dipping outcomes. Then if we look at the relationship between overnight blood pressure and our sleep timing variability, so we have the standard deviation of sleep onset timing here on the x-axis, we see that there was no clear relationship between the variability of sleep onset and our blood pressure in our day shift workers. When we look within the night shift workers, not only is there higher variation in their sleep onset timing, but now this is significantly related to poor blood pressure dipping outcomes. So potentially the more variable that sleep schedule is, may be indicative of more circadian disruptions, which are therefore negatively impacting these blood pressure profiles. And so taken together, this body of evidence demonstrates that Shift work does disrupt blood pressure dipping patterns. This happens quickly when transitioning into a new job, and it is sustained even after years of working in a shift working occupation. And that these blood pressure dip outcomes appear to be associated with disruptions to the circadian timing system. So that's kind of an extreme example, but what about mild and potentially chronic disruptions in non-shift working, otherwise healthy adults in real world settings? So as it turns out, there is a range of our internal circadian clock timing. So here's just a representative sample where we have the length of the circadian clock here on the x-axis, and these are a number of participants here on the y. And we can see that a majority of individuals fall outside of that 24-hour range. So their internal circadian clocks are ticking a little bit longer than the 24-hour Earth's rotation. So what that means is a significant number of people likely have to adjust or advance their circadian clocks every day to match with our 24-hour Earth's rotation cycle. And so potentially experiencing reoccurring, small but reoccurring misalignments between their internal clocks and their daily behaviors. And so some work by Kelly Barron and colleagues were interested in seeing how circadian alignment related to body composition and metabolic health markers. And so first they looked at these relationships in healthy weight individuals. And what they found was that there was an inverse relationship between circadian alignment and body fat percentage and abdominal body fat distribution. But this was only seen in their male participants. These relationships weren't observed amongst their female participants. Then in a follow-up study, they looked to see the relationship between circadian alignment and metabolic health. And what they found in their overweight and obese individuals, there was this inverse relationship between circadian alignment and insulin resistance, but only observed with the individuals with overweight and obesity. These relationships were not seen in the healthy weight individuals. So taken together, these findings would suggest that the effects of circadian misalignment on these cardiometabolic health outcomes might be differentially affected by sex, and that these negative effects could be more acute in an overweight and obese phenotype. And so that's what was the background that led to sort of my next question is, are there sex-specific differences in these cardiometabolic health outcomes in otherwise healthy adults with overweight and obesity, just in real-world settings, and to see what those relationships look like. So in this cross-sectional analysis, we had 30 participants, 15 of which were female. These consisted of a sample of newly hired nurses and bus operators. There was no self-reported history of hypertension, diabetes, sleep disorder, pregnancy, nor had anyone worked an overnight shift within the previous 3 months. So I do want to stress that this data all came from individuals who were on a day-oriented working schedule. Our in-laboratory measures included an assessment of body composition, and this was done with dual X-ray absorptiometry. Here's an example of the printout here. With this scan, we get a marker of BMI, which is just a weight to height ratio. But we also get a marker of total body fat percentage. And then we also get an android-gynoid ratio, which takes the proportion of abdominal body fat distribution relative to the amount of body fat distribution in the waist and hips area. So a higher number, a higher AG ratio, would be representative of greater abdominal body fat distribution. And this is important because higher abdominal obesity is associated with poorer metabolic health outcomes. Next, participants had a fasted blood draw where we assessed blood lipid profiles. Circadian phase was again assessed by dim light melatonin onset. And then similarly, patients reported self-reported sleep onset for a week after the visit. Phase angle of entrainment was again calculated the same way as the time difference between DILMO and average sleep onset, but we took it a step further this time for this analysis. So I had mentioned previously that a longer duration between these two time points would be considered a wide phase angle. But if these two time points were now occurring closer together, this could be considered a narrow phase angle. And so what we did was we categorized participants into wide or narrow phase angle groups based off of the median split within this sample. And then we calculated a metabolic syndrome risk score, which is just a composite score consisting of waist circumference and height, fasting glucose, triglycerides, and high-density lipoprotein, and resting systolic blood pressure. So looking at the characteristics between our female and our male participants, we see that there was no differences between sexes for age or BMI. However, females did have significantly higher levels of overall body fat percentages, whereas males had higher AG ratios, so potentially representative of greater abdominal obesity. Our male participants also had significantly higher levels of circulating triglycerides and significantly higher metabolic syndrome risk scores. If we look at our sleep and circadian phase variables, we see, apologize for that being not aligned properly. We see that there was no differences between sexes for dim light melatonin onset, for average sleep onset, average sleep duration, or general phase angle. So there was no differences between the sexes for phase angle. And then if we look within our phase angle groups, similarly, we did not see any differences between males and females within the narrow phase angle group, nor in the wide phase angle group. So here we are looking at our key body composition metrics, starting with BMI first, which is here on the y-axis. On the x-axis, we have our narrow and wide phase angle groups. And in the left-hand column, we'll see data for our female participants, and in the right-hand column will be data for our male participants. So looking at our female participants first, there was no differences in BMI between the narrow or the wide phase angle groups. If we look at overall body fat percentage, here's where we see that females within the narrow phase angle group had significantly higher levels of overall body fat percentages compared to females in the wide phase angle group. But if we look at our android gynoid body fat distribution, we see no differences. So overall levels of body fat percentages higher, not explicitly located within the abdominal region. Now, if we look at these markers in our male participants, similarly, we see no differences in BMI between the narrow and wide phase angle within our males. No statistical differences in our overall body fat percentages. However, we do see that males in the narrow phase angle group had significantly higher android gynoid ratios, so higher levels of abdominal body fat distribution compared to males in the wide phase angle group. And so while there's no differences in BMI, just that crude measure of weight versus height, we do see that there are sex-specific differences in body fat distribution that appear to be mediated by the circadian timing system. So if we look at our groups again, but this time with our cardiometabolic health markers, we have glucose here first. Looking at our female participants first, we see that females in the narrow phase angle group had significantly higher levels of fasted circulating blood glucose compared to females in the wide phase angle group. Here we have our triglyceride levels, and no significant differences within our females between these groups here. And if we look at our metabolic syndrome risk score, similarly, we see no differences within our female groups for metabolic syndrome risk. Looking at our male participants, we see no differences here for circulating glucose. However, males in the narrow phase angle group had significantly higher levels of triglycerides compared to males in the wide phase angle group. And they also had significantly higher levels of the metabolic syndrome risk score. And so these data would suggest that the circadian effects on metabolic regulation may be differentially affected by sex, which potentially, females predominantly favoring carbohydrate or glucose regulation, males potentially tapping into more lipid regulation for metabolic effects. Although more work needs to be done, it is quite interesting. So then we put together a correlation matrix looking at the relationships between phase angle and our key markers of interest, including body fat percentage, our android gynoid ratio, heart rate, triglycerides, and our metabolic risk score. If we look at the data within our female participants first, what we see is that there is a significant and negative correlation between phase angle and body fat percentage and resting heart rate, such that a narrower phase angle was associated with overall higher levels of body fat percentage and elevated resting heart rate levels. And interestingly, different relationships emerge when we look within our male participants, such that a narrower phase angle was associated with higher android gynoid body fat distribution and a non-significant trend towards elevated triglyceride levels. So taken together, it would seem that sleep and circadian disturbances do impact cardiometabolic and body composition outcomes differently by sex, which could provide really interesting opportunities to do like sex-specific targeted approaches to promote overall health and well-being. So this led me to ask sort of my next question, well, are there sex-specific differences with circadian disruptions with markers of inflammation? And I was particularly drawn to this question because there's been some compelling evidence and some recent evidence looking at inflammatory marker C-reactive protein in healthy women across the lifespan. So in otherwise healthy women, if you have elevated levels of C-reactive protein, this is predictive of future cardiovascular risk across the next 30 years. And so I thought, okay, well, if we could maybe see if there's some sort of sleep circadian disruptions that's affecting these markers of inflammation, that could be a target that we could use to help promote overall health. So this is just a subsample from the data set that I had showed previously. We don't have C-reactive protein data in every single participant, but here's what we had. So we have C-reactive protein level, again, our global marker of inflammation here on the Y, only a subset of 9 females and 10 males. And again, these are all participants, healthy participants with the overweight and obesity. If we look at our females in the narrow phase angle group, on average, they had significantly higher levels of C-reactive protein compared to the female participants in the wide phase angle group. And if we look at the relationship between CRP and phase angle, there is a non-significant trend towards narrower phase angles being associated with elevated levels of CRP. But if we look at these within our male participants, we see no discernible differences in CRP between the narrow and wide phase angle groups, and no obvious relationship between phase angle and this inflammatory marker. So that led me to wonder, what could be some of the mechanistic contributors that are underlying these findings? Because I don't think it's phase angle per se. I don't think it's the time difference between DILMO and sleep necessarily that's driving these relationships. But rather, phase angle may be a biomarker of downstream contributors that are altering mechanistic pathways that affect health. I'll kind of go into what I mean by that. So in this subset of individuals, I have our circadian and our sleep variables. If we look between our two groups, the females in the narrow phase angle group, non-significantly, but tended to have later DILMOs and tended to go to bed earlier compared to the females in the wide group, not surprisingly, depending, given how we calculated phase angle. But they also tended to wake up earlier than the females in the wide group. If we look in this subset of male participants, we see a similar trend that males in the narrow phase angle group tended to have later DILMOs, and again, tended to go to bed earlier. And there was no discernible differences between wake time between the two groups, but what kind of jumped out at me is that 5 a.m. seems like an early clock hour, right? And it's early, especially compared to the females in the narrow phase angle group. So that led me to wonder, well, could it be that these individuals are waking up early and potentially experiencing some sort of morning circadian disruptions or morning circadian misalignment where now they're awake maybe before their internal biological clock is ready? And this isn't just our hypothesis, others have also speculated on this. And I do want to mention that we don't have dim light melatonin offset in these participants, so the point at which melatonin goes down and stays down. And so we can only speculate if these participants are beginning their waking activities when melatonin might still be high and that they might be experiencing these disruptions. But I wondered, is there a way that we could try to target this? Is there a way that we can try to look if they might be experiencing some sort of disruptions occurring in the morning? And I thought, well, we looked at the standard deviation onset in our shift working population, trying to see how variable is it that you go to bed on a day-to-day basis, trying to target, if you're experiencing some sort of disruptions in the evening. Could we look at something similar with wake onset? Could we look at the standard deviation of wake onset and see, is there a lot of variability in the time that you're waking up on a day-to-day basis? So here we have the relationship between C-reactive protein and the standard deviation of wake onset. For this analysis, just because I was interested and really wanted to see if anything would show up, I've added data from some of my currently ongoing projects that are funded through the Medical Research Foundation of Oregon and the Sleep Research Society Foundation. And so this was just as much CRP data as I could find. So you can see we have 36 individuals in this analysis, 18 of which are females. And this is everybody, right? This is healthy weight. This is individuals with overweight and obesity, just to see if we see anything there. And so when I plot our female participants first, we do see a relationship between the standard deviation of wake onset. So the more variable you are when you're waking up is associated with higher levels of C-reactive protein. And if we look at these relationships within our male participants, we don't necessarily see any relationship there. Although, this is super interesting. This is some of my newer data that I've been playing with. So I do want to caution, I do think these should be interpreted with some reservation, because I don't think the effect of overweight and obesity should be understated. I do believe that there is an interplay between these sleep mechanisms, overweight and obesity, and some of these health markers. And when we look at this relationship between wake onset variability and CRP just within our individuals of overweight and obesity, we don't see a significant trend, although it does look like it's maybe moving in the right direction. And then again, looking at our male participants, no relationship, if not the opposite relationship. So the effect of morning circadian disruptions on markers of inflammation remains a bit unclear. Might be a function of a lower sample size and a more heterogeneous population, but this is something that I do want to continue to think about, maybe pull on this thread as we collect more data and as my thoughts really start to formulate. But I was wondering, okay, so maybe we don't see anything with markers of inflammation. Are there other key vascular health markers that we could look at that may be related to this morning circadian disruption? And so given the importance of overnight blood pressure dipping for predicting future cardiovascular risk and mortality, I was interested in seeing, okay, do we see any relationship between wake onset timing and our blood pressure dipping? Similarly with the CRP, I collected as much data as I could find, threw it all into our spreadsheet. So this is 51 participants, 34 of which were females. And what we see is that the higher the variability is on when you're waking up, the poorer your blood pressure dipping outcomes appear to be. A relationship that is not observed within the male participants. So potentially an effect of morning circadian disruptions on blood pressure dipping while we sleep within females, maybe not so much in males. But then I wondered, okay, is this reflective of morning specific disruption? Is this just general disruption? And so, should I be fair and assess these relationships in relation to the standard deviation of sleep onset timing? As you may recall, we did see these relationships within our shift working population. Greater variability on when you're going to bed at night was associated with poor blood pressure dipping in our chronic night shift workers. Do these results kind of show similar patterns in real world non-shift working settings? So I plotted the males first. Apologies, because I've been showing you female data first this whole time, but I wanted to show that male participants, we do see that there's this association between greater sleep onset timing and poor blood pressure dipping, although, admittedly, it looks like it's getting pulled by a few people who are very variable and have poor blood pressure dipping outcomes. But when we look in our female participants, we don't see any sort of relationship between when they're going to bed and what that might be, how that might be related to their blood pressure dipping outcomes. And so I just wanted to take 1 second to remove some of the non-significant noise and just look at these significant relationships. One within our females related to the standard deviation of wake onset timing, so how variable it is when we're waking up, versus the males of how variable when they're going to sleep in these relationships to blood pressure outcomes. And like I said, these are the newest analyses that I've sort of embarked on. Like, this is hot off the press. This is just last week, I kind of went down this rabbit hole. And so I'm super excited about this. I feel like this might indicate that potentially it depends on when in the circadian phase these disruptions are occurring, that might be contributing to poor vascular outcomes, and that these might differ between men and women. And so I have added a question mark here that potentially morning alignment and vascular health might be affected by sex-specific effects. Again, still trying to like put my head around it, think about it. I thought this would be a great forum to debut these data for the first time. So if anybody has any thoughts or objections, I'm super happy to talk about that. But I do think that this could be a key step in informing sex-specific approaches where we can try to promote sleep and circadian health to promote overall well-being specific towards men and women. And then last but not least, we are interested in looking at the effects of circadian misalignment on health outcomes in clinical conditions. So this is the most recent data that we have under review with JACC Advances. This is looking at the influence of circadian disruptions on adults with heart failure. And so heart failure hospitalizations is a growing epidemic, and individuals with heart failure often experience a lot of sleep difficulties. What's even more interesting is that in rodents, if you make them cardiomyopathic and then you alter their sleep-wake opportunities to induce an irregular sleep pattern, and if you cause circadian disruption, that significantly reduces lifespan. And so the regularity in our sleep habits is really important for longevity. And there's even epidemiological evidence in the general population that has shown that sleep regularity, how regular you are on a day-to-day basis, is better at predicting overall mortality than sleep duration. And so there's something key to that regularity in sleep habits. So with this project, we have collaborated with Dr. Quinn Denfeld in School of Nursing. Her work really focuses on frailty in adults with heart failure. And so together we combined our heads and we sought to look at how these sleep patterns affected clinical event risk across a 6-month period. And so in this sample, there were 32 participants, 14 of which were female. All participants had confirmed heart failure diagnosis of at least 3 months, and they were recruited following a hospitalization for acute decompensated heart failure. And so hospitalization resulting from a sudden worsening of signs or symptoms related to heart failure. And this is a really critical moment for these patients because oftentimes after this hospitalization, the rates of rehospitalization are elevated. And so this would be a really great time to try to have some sort of interventional target. So our baseline measures included demographic and clinical data taken from surveys and the electronic medical records. This included heart failure severity, heart failure type, medications, et cetera. These participants were also given sleep diaries for self-reported sleep onset, which they filled for the week following hospital release. So this was their sleeping while they were at home. And then we tracked clinical event data through the next 6 months. And so this included all-cause emergency room visit, hospitalization, and death. And so our calculated measures were the sleep regularity index. And this is the probability of being awake or asleep at any 2 time points hours apart. So here in this diagram, these hash boxes indicate a sleep episode. So what's the likelihood you're awake or asleep at this time point, or this time point, or this time point, or maybe this time point? And so this is a bit different than the standard deviation of sleep onset or standard deviation of wake onset, because this really encompasses the whole 24-hour cycle. It can capture the effects of naps, so not just your main sleep bout, but if you are taking little naps throughout the day, which is quite frequent in this population. So a bit more of a robust measure. And then based off of the sleep regularity index, we categorize participants into regular sleepers, defined as greater than 87%, or irregular sleepers, which was less than or equal to 87%. And then we calculated a Seattle Heart Failure Model score, which estimated heart failure severity, but also is a percentage of likelihood of survival over a course of a specific period. So here we have our participant characteristics between the regular sleepers and the irregular sleepers. There was no differences in age or BMI. However, irregular sleepers did have a higher Charlson comorbidity index, which is just a weighted index to assess overall comorbidities. And there was no differences between groups in the prevalence of obstructive sleep apnea, which is often common in this disorder. Our heart failure characteristics included the duration of heart failure, so no differences there for how long they had been diagnosed for heart failure. No differences in the prevalence of non-ischemic etiology or heart failure with reduced ejection fraction. And there was no difference in this Seattle heart failure model 1-year projected survival percentage. For our sleep characteristics, there was no differences in the PROMIS self-related sleep impairment score. And so this is just a questionnaire that assesses how do you subjectively feel your sleep has been in the past week. No differences in total sleep duration. No differences in sleep onset. And then by design, the participants in the irregular sleep group had significantly lower sleep regularity indexes. So we created a Kaplan-Meier time to event curve, and this included all the events that we had calculated across that 6-month period. And so on the Y, we have event probability, with the top here being 0%, the bottom here being 100%. And on the X, we have time from hospitalization days. So this was discharged from that baseline hospitalization day across the 6-month time period. And when we look at the event risk in our regular sleep group, we do see that over time, there is a risk for a reoccurring event or maybe a reoccurring hospitalization. But what was really striking is when we compared this risk to the irregular sleep timing group, there was a significant difference between these groups. And so the risk of an event was more than twice that in the irregular sleep group compared to the regular sleep. Not only did the sleep regularity index predict the 6-month event risk outcome, but it was more than twice the risk compared to the regular sleep group. And this remained significant even after we controlled for that Seattle heart failure model. So very striking. We have this under review right now, so fingers crossed that we get some really good feedback, because this could provide like a really great non-pharmacological behavioral target to try to not only mitigate, adverse cardiovascular, future cardiovascular events, but try to reduce the risk of rehospitalization as, following that like acute decompensated heart failure event. And so in summary, we did find that irregular sleep timing did predict worse clinical events in this population, opening up a lot of doors for potential targets. So in summary, this body of data working on the last few years has shown that sleep and circadian disruptions contribute to negative metabolic and cardiovascular health outcomes. We see that these adverse health effects occur in shift working occupations. They happen quickly. They are sustained. There also appears to be sex-specific differences in the effects of these sleep and circadian disturbances between males and females, again, potentially opening up the door to sex-specific approaches to promote health. And then in clinical conditions, particularly amongst adults with heart failure, the regularity in your sleep habits and sleep patterns are really key to trying to maximize health over the long term. Some of my currently ongoing projects I had mentioned earlier, because I've added to those earlier data, I'm currently looking at the impact of sleep timing regularity on cardiovascular, metabolic, and autonomic function. We're interested to see if you are irregular and we make you more regular, can that help with some of these underlying health mechanisms? So that project is underway. I'm also interested in looking at the effects of sleep timing regularity on proteomic biomarkers associated with cardiovascular health. And so is there like a plasma protein marker that we can find that maybe could be suggestive of some of these underlying mechanisms that we could target? And so this work is funded by the Sleep Research Society Foundation. And then some of my future projects include looking at the impact of circadian misalignment on sex-specific mechanistic pathways that affect cardiometabolic health. So for this project, we'll be able to leverage a natural occupational experiment that occurs when newly hired nurses are brought on. They are onboarded and trained during a day-oriented schedule. And then the majority of them end up getting scheduled for a night shift schedule. And so that's like a really rapid 12-hour shift in their sleep-wake behaviors. So based off these data and these findings, I'm really interested in getting in and seeing like how these effects might differ between men and women. And as Andrew had mentioned at the beginning, I was recently appointed to the Oregon Birch K-12 program, and so that will be supporting these research endeavors over the next couple of years. And it's really great and helpful because it'll allow some support for myself while I attempt to secure longer-term NIH funding. So I'll be resubmitting my KO1 submission this summer with revisions. So fingers crossed there. And with that, I would like to acknowledge my mentor, Dr. Andrew Mackhill, all our co-collaborators, the Sleep Chronobiology and Health team, the majority of which are here today, and then our funding sources that have supported these endeavors. But particularly, I'd like to highlight the T32, which has supported me in these last 2 years, and then Medical Research Foundation and Sleep Research Society. And with that, I'm happy to take any questions.
So out of curiosity, so, I mean, the light obviously affects us better. So are there any indications that, for example, you know, in summertime, especially also if we have to shift to the normal, that they are more heart failures in like the middle of summer or the middle of winter when our internal rhythm is off from the night rhythm?
Okay, yeah, great question. The question for those online was, are there, if I can summarize, please tell me if I summarize it correctly, are there seasonal effects in rates of heart failure given the differences between like the light exposure and like the shorter days in the winter versus the longer days in the summer? I don't know the answer to that per se, but my guess would be potentially not, just because with the invention of electrical light, we can sort of mask any sort of seasonal effects we might have on our circadian timing system, because now we're able to get exposed to lights whenever we want, just like with a flip of the switch. So my guess would be possibly not, but I'm not entirely sure.
But for light, I mean, in summer you get a lot. I mean, hardly anyone completely lives in the dark.
Yeah.
So, I mean, that one, I mean, you can regulate better switching light on, but switching the daylight off is harder.
So the light, so you're saying the light at night, because of the longer summer hours, might be more.
I mean, like, if they are real summer, with the daylight shift, then it's fine. It's still light out there at like 9:30,
Sure, yeah.
which, you know, would not be fit with our sleep pattern, because our melatonin should go up at that time, start going up. But if the light is there, it's not. Or it's not, I guess.
Yeah, and well, the question is the summer though. So, oh.
So I mean, my turning clock is not shifting with that, right? But the outside.
I
So
think.
that's a mix, like what I thought that should be a mix of nine and 15.
But we're still controlling right now. So even though the sun goes down, it's still like
And it's how much?
it's the summer, the light. So we're not changing the light at night. So yeah, the winter is more different. It starts up earlier. And then in the summer, we still do light.
And in the morning, I mean, I have to admit, I mean, my way, for example, in summer, I get light at like, you know, 4 in the morning, but I can do that later. Even if I have preference for it, I guess. Also,
It might be, we don't see those in the summer.
it was hard for me also, if there's some clinical data that they are changing this hard for the
It's good. Yeah, I don't know. It's a good question. Yeah. Oh, sorry. Steve, go ahead. You.
I just wondered if the sleep regularity index goes along with eating irregularity as well. So if you sleep in a regular fashion, you eat in a regular fashion, is that expected to get that?
Yeah, so the question was if the sleep regularity index would follow along with irregular eating patterns, and if that could also contribute to some of these effects. I would say it's possible. I don't have as much of a background in like this clinical demographic specifically, so I don't know if just like even amongst regular participants with heart failure, what their eating patterns are like, if their appetite is altered as a result of like their heart failure and some of like the symptoms that they're experiencing. And so my guess would be if you are, sleeping more hours of the day, you're not eating, or maybe you're eating at times that are not aligned with your circadian system, which could certainly have an impact. But I don't think we have any eating data in this population, but it is a good thing to think about.
Along those lines, one of the interventions you maybe want to recognize sleep.
Yes, it does sound easy, and it's funny because so we have embarked on this for maybe 9 or 10 months, and we've gotten 3, almost 4. I think we've had 4 people fully complete the intervention. So honestly, the hardest part with this study is finding irregular sleepers. I think people are more regular than they realize, well, the one that we have currently ongoing, I'm not sure what our average age is right now. I'd say it's about, people on average probably in their 30s, predominantly women so far that are interested in this study. And so what we do with this is if we identify somebody who has a pretty irregular sleep pattern, we track them for a couple weeks to try to get a better idea of their habitual habits. Then we ask them to just pick a bedtime that works best for them most nights during the week and try to go to bed within 30 minutes because we think it is more of the consistency that is impactful for health markers. And so it can be whatever time, if 11 p.m. works, try to go to bed between 10:30 and 11:30. I think one of our participants picked 1 a.m. And so he was like between 12:30 and 1:30 worked for him. Yeah, the hardest part with that is finding irregular sleepers. But so far, we've had success, at least in making them regular and keeping them regular. Whether or not that is translated to different findings, we haven't started digging into those analyses yet. Fingers crossed, yeah.
So going to your measurements of the blood pressure, is there an individual participating in that study that is on pharmacological blood pressure medication? Have you collected data that would suggest that individuals that are on regular daily blood pressure medication is going to have a different magnitude of change related to that fluctuation or for those who are on medication, are they being excluded from the study?
Yeah, so the question was, how would medications affect the 24-hour rhythm of blood pressure and the magnitude of the dip? For these data, we did exclude for medication use, and so I'm not entirely sure what the literature would look like for individuals on hypertension medication. I mean, if they are successfully treating their blood pressure, I would hope that it would go normalize their blood, like their 24-hour blood pressure profile. Although I know that there can be some trickiness treating hypertension. Not everybody's responsive to the medications. They might need to have multiple medications to try to treat their hypertension. And so that would be like a whole nother interesting thing to look at. Yeah. Yeah.
So I thought it was very interesting, the data of women waking up early in the morning and they're still have melatonin. And have you dug into why you think and the health effects of like jumping out of bed and looking at your phone and starting working out?
Oh,
Or
yeah.
being interrupted by someone who makes food in the morning, and how that, what do you kind of get that?
Yeah, well, so the question was in the data with the women having irregular, wake timings, how to target if they're like getting woken up for, family or personal obligations, if they're waking up and they're starting work right away. And like, I know for me personally, I've got not great morning habits. Sometimes I wake up. So because I thought about this from an anecdotal standpoint, like sometimes I wake up really early if I want to get after it at the gym. Sometimes I wake up a little bit later if I want to drink my coffee on the couch and doom scroll the news. That's a great way to start the day. And then sometimes if I don't sleep well, I can maybe push my alarm clock until I really need to get out of bed and get ready to go to work. So my sleep waking can be quite variable. I think the first step is to look at this data maybe a little bit more critically. Like I said, this hot off the press within the last week. And so I do want to make sure that like I'm thinking about all the other variables that could be affecting these relationships. But if it is something that's a promising avenue, I think the next step would be trying to, maybe more directly target if they are, waking up during when the melatonin levels are high, seeing when DILM off is, if there are these relationships still present, and then maybe working from there to see, how could we, what are the best practices, best recommendations for that. So I'm just super pumped about that. So we'll see. We'll see. Ask me again in like a year. Yeah, and that's a great question. Sorry, for those online, the question is, if I could potentially summarize it in the proper way, if the heart failure severity or the severity of like the decompensation event could be affecting the sleep regularity, as opposed to the sleep regularity, being a bit more related or driving, I think, these outcomes. So the marker of the heart failure severity that we have is that Seattle heart failure model score. And there was no differences between those two groups for that. When we looked at the models unadjusted, so not throwing in anything, we saw these significant differences between the groups. And then even when we added that score into the model, which from my understanding is a common marker that you would add as like a covariate, we still saw those significant differences between those groups. We couldn't add a whole bunch of stuff just because of the number of participants that we had, so we were a little selective with what we were looking at and controlling for. So from my understanding, in terms of like heart rate severity, they were similar between the groups. But sorry, I know you. I think one of the targets is like hopefully trying to see, because sleep is probably disrupted during the hospital stay. How does that then translate into disrupted sleep at home? Now all of a sudden there might be new medication regimens, a lot more things that they have to incorporate into their day-to-day life, and so it is sort of a stressful time. So whether that is also partially driving the irregular sleep, it absolutely could be a factor. Yeah. I am not 100. So my understanding, I think it's like more of a global assessment, and it's a predictor of survival and longevity, and you can have it at different time points. So we showed you data from, survival for the next 1 year. You can also extend it up to 5 years. You can also extend it up to 10 years. But I would have to go back and double check how it's specifically. We worked with the collaborators in Quinn Denfield's lab for these markers, so I'd have to double check specifically. Thank you. Oh. Great. Yeah, thank you.
A lot of stuff to get through.
Yeah, thank you. Hi. Oh my gosh, it's so nice to meet you in person. No, you're fine. You're fine. I was already in the group, so I didn't even notice.
Okay, good. Fantastic. Okay, great job.
Thank you.
We'll talk soon.
Yeah, absolutely.
Was that the alarm?
Oh, there's another class.
Oh.
We got two.